In this recording, I'm going to introduce you to Lab 27, non-vascular plants and seedless vascular plants. Our non-vascular plants are the mosses, which carpet the forest that you might walk in. They're very small, low to the ground. And our seedless vascular plants, we'll be looking at ferns. So you should notice that ferns have spores, not seeds. As your lab indicates, your learning goals discuss the evolution and diversity of land plants. So we'll be starting with green algae, the common ancestor to land plants, and the transition from water to land, that the mosses, our most primitive land plants, moving to ferns, pines, or conifers, and then angiosperms, or our flowering plants. So we'll be looking at that. We're going to look at the concept of dominant generation in plants. They have two generations. We'll look at dominant generation. We'll spend some time on the mosses, again, our non-vascular plants, how they're adapted to land, and the appearance of two generations. And we'll also be looking at ferns and how they adapted to reproducing on land, and again, the two generations. Let's start out by looking at a phylogenetic tree, a simple phylogenetic tree, to show the evolutionary relationships. Okay, so right here we see the common ancestor, green algae. Okay, not red or brown, but green algae. And you know, the common ancestor most likely could survive periods of desiccation or drying out as it lived in the water. But what was life in the water like? There wasn't consistent desiccation, so there may be periods of drying out, but basically water was not a problem. Water supplied the green algae with buoyant support. Just think of seaweed. Seaweed outside the water is just floppy. Water provides buoyant support for the plant. Their gametes, egg and sperm, can be carried away by water currents, and the embryo stays moist. So life in the water definitely had its advantages. So why did organisms move to land? Well, on land there's ample sunlight. Okay, all waves of sunlight, none are blocked by the water, so including the red and blue. So there's more sunlight on land. Early in evolution, there were no herbivores, so there's nothing that's going to eat the plants. Okay, so how do scientists know that green algae was the common ancestor? Well, just like plants, green algae have a cell wall made of cellulose. Just like plants, green algae store carbon as starch. And just like plants, green algae have chlorophyll B and A and similar DNA and RNA to land plants. So again, the most primitive of the plant families or plant groups are the mosses. They grow low to the ground and in a moist environment. With the evolution of our vascular tissue, the ferns can grow a lot taller because vascular tissue, as we will learn, gives structural support as well as a means of getting water from the roots up to the top part of the plant. Next, we have the evolution of seeds that further protect the embryo. And in the next lab, we'll be looking at conifers or cone-bearing plants. And in the next lab, we'll also be looking at angiosperms or flowering plants. So this more detailed phylogenetic tree or evolutionary tree can be found on page 358 in your lab book. So we'll notice along the x-axis is millions of years. So we're going back to 550 million years ago. Remember that the Earth is dated to 4.5 billion years. So 550 million years ago up to the present time. And on this axis, we see the algae, the common ancestor, which this would be an outgroup. And we see our non-vascular plants called the bryophytes, with our hornworts, liverworts, and mosses. We're going to focus on the mosses. Then we also see the seedless vascular and seeded vascular plants. And in this lab, we'll look at the ferns. So as we look at this tree, again, this is our outgroup without any of these derived characteristics. So embryo protection is the first derived characteristic that we see. So embryo protection, something to note is that plants are called embryophytes, phytes for the plants. They retain their embryo in the gametangium. Okay, and in our case, the gametangium produces gametes. There's male and female gametangium. The embryo is maintained in the archegonia. That's the female gamete producing structure, and there's a moist chamber so that the embryo does not dry out. So all of the groups from here on up have some type of embryo protection. Another adaptation is apical growth. So as we move from above this group right here, the bryophytes, all of these groups have vascular tissue and also apical growth. So they have roots and shoots. And the roots and shoots contain basically a region of stem cells. Just see, I think I have this. A region of cells that can become any type of plant tissue like our stem cells. So for plant tissues, as we'll learn later, plants have vascular, ground, and dermal tissue. So the evolution of apical growth allowed a lot of primary growth. Plants could get very tall with these types of cells. Something that's not mentioned in here is that in addition, plants also evolved cuticle, waxy cuticle to keep from drying out. All right. Here we see the evolution of vascular tissue. So vascular tissue, basically there's xylem and phloem. So xylem allows water to be transported from the roots to the leaves, and then phloem is vascular tissue that brings nutrients from the leaves to the roots. And vascular tissue also provides support for plants, allowing them to grow tall. Right here we're looking at megaphylls and microphylls. So in your textbook there's a nice picture of this. So microphyll leaves have one vein of unbranched vascular tissue, and those are our lycophants here, or club mosses. Megaphyll leaves have branched vascular tissue, and that is your fern, allowing them to spread out more, capture more sunlight. And then we see the evolution of seeds. So important things about seeds. Seeds contain the embryo, and the seed can remain dormant until favorable conditions for growth arise. So seeds can also be transported by wind and water or in the feces of animals. Okay. And then finally, the flowering plants or angiosperms have fruit to make those seeds appealing to animals. So animals will eat the fruit and then disperse the seeds in their troubles. So next, what we want to do is take a look at alternation of generation. So all plants have an alternation of generation. And what that means is that there are two multicellular forms. There's the sporophyte generation and the gametophyte generation. The sporophyte generation produces spores by meiosis and is diploid, 2N. We're going to take a look at this in more detail. The gametophyte generation is haploid and therefore N, one set of chromosomes, and it produces gametes by mitosis, which sounds odd. We're going to explore that in just a minute. One goal of your lab report, of your lab, is to recognize which is the dominant form, the sporophyte or the gametophyte. And with moss, the gametophyte generation, so the haploid generation, is actually the dominant form, meaning that is what you would see. So when you walk through the forest, if you see a lot of moss, you're looking at a haploid structure of moss. So, you know, like our egg and sperm are our haploid cells, but with moss, you have a multicellular organism. You've got the whole being is haploid. And the sporophyte is the not dominant generation, or the one that's more difficult to see. And mosses actually have their diploid generation, which would be like our body, but they're living off of, the sporophyte lives off of the gametophyte generation, or is dependent on. With each of the other groups, plant groups, the dominant generation is the sporophyte generation. Okay, so the 2N generation. So your fern leaves are your dominant generation. The gametophyte is this little prothallus at the bottom that's about ½ centimeter big. Our trees and our flowering plants are also sporophyte dominant. So let's explore the life cycle of the moss right here. And this picture can be found in your lab manual on page 360. Let's see if I can this over just a little bit. Okay, so with moss, we're going to start right down here with the structure of moss that we can see with the gametophyte generation. So the mature gametophyte, that what we think of as moss, is haploid, and there's actually male and female moss plants. So the male moss plant produces sperm in the antheridia. The female moss plant produces eggs in the archegonia. What happens is fertilization will occur. There needs to be a rain or at least a lot of water to move these flagellated sperm over to fertilize the egg. So these are haploid. Once fertilization occurs, we have a diploid zygote. So the zygote is going to grow within the archegonia, within the female structure, because it's protected from drying out. So by the process of mitosis, the zygote develops and grows into the sporophyte. The mature sporophyte lives on top of the moss plant as this stalk right here. So this is our sporophyte generation, and this is our diploid generation. On the sporophyte, we see right here that we have the sporangium, okay, and this is where the spores are going to be produced. And the spores are haploid. The spores are produced by meiosis. When the spores, under the right conditions, they erupt from this structure and they're carried by the wind to a new location. And again, the spores are haploid. The spores become the protonema, which is the precursor to the gametophyte, and then we start our life cycle over again. So we have haploid and diploid structure. So to summarize, with moss, our haploids are the gametophytes, the spores, the sperm, and the egg. Our diploid structure is the developing zygote, which becomes a sporophyte. Before we end, let's take a look at the ferns and their generation. So the ferns, again, have the gametophyte and the sporophyte generation, but with ferns, the gametophyte is this small, heart-shaped prothallus, about 1/2 centimeter big. You probably don't even see it. With the sperms, I'm sorry, excuse me, please. With the ferns, the sporophyte generation is large. That is a structure that we see, and we will look at the sorus, which contains the spore-generating sporangium, which makes the spores. This is a picture of our fern life cycle. Here's the gametophyte. This is the prothallus, and the underside contains the antheridium and archegonium. The antheridium makes the flagellated sperm, and the archegonium produces the egg. Under moist conditions, the sperm meets the egg, fertilization happens, we get the zygote, and then through mitosis, we get the young sporophyte, which grows on top of the gametophyte. So there's your sporophyte. The sporophyte becomes the fern, and you'll have a chance to look on the underside of some species of ferns, having trouble with that word, ferns today contain sori, okay? The sori contain sporangium, which generate spores. The spores then are haploid. They're dispersed by wind currents, and then the spore will germinate to produce the gametophyte, and the life cycle continues over and over. So please check Canvas for the lab report, and I hope that was helpful in getting you started with your lab.